welcome everybody to today's webinar, and thank you all for taking the time to hear about our project HemAcure. The structure of this webinar is the following. After a short introduction, who we are and why we are here, we want to shortly outline the background of the bleeding disorder Haemophilia A and the current treatment and its history. Afterwards, we introduce the workflow of our project, and all principals, investigators of the teams have the time to report about the reports over the past 3 years. At the end, we want to take a glimpse into potential future projects, and we have prepared some answers to the frequently asked questions section, which you raised in the course of the registration to this event. Of course, you can text us via the Q&A section on top of the screen, and we will try to get back to you during this webinar session. Nonetheless, we probably won't be able to answer all questions. This is why we decided to write a follow-up in which we answer all the questions in all 3 languages the consortium is built of: English, German, and Italian. One more information before we start: we do record this webinar, and we will upload it afterwards to Arctic's YouTube channel and the HemAcure webpage, so that you can re-watch it and share it with your social network. Let's get started. This is us. We are the HemAcure team. We are an international group consisting of people from different nations working in Italy, UK, Canada, and Germany. Together, we form the consortium working on this EU-funded Horizon 2020 project, HemAcure. To start off the scientific part, we want to show you a video generated by CSL Behring, which very nicely explains what hemophilia is and how it is currently treated.
So, what is hemophilia? Hemophilia is a bleeding disorder, a problem with a person's blood that makes them bleed longer than a person without hemophilia. No one can catch hemophilia. It's an inherited condition you have from birth, for your whole life. It's just part of who you are, like the color of your hair or the shape of your nose. Here's how it works. Your blood travels through your body in tubes called arteries, veins, and capillaries. Normally, when there's a tear or hole in one of these, bleeding starts, and different parts of the blood go to work to plug the hole. Blood cells called platelets gather at the opening, and other blood parts called clotting factors help hold the platelets together to form a clot that stops the bleeding. When a person has hemophilia, he doesn't have enough of one of those clotting factors he needs to form a clot, and he keeps bleeding longer than he should. Each person with hemophilia is missing one of 2 kinds of factors. If you're missing factor 8, you have hemophilia A. If you're missing factor 9, you have hemophilia B. Either one can be mild, moderate, or severe. Without regular treatment, having hemophilia means you'll bleed longer than you should if you get cut. But you don't have to have a cut to have a bleed. Sometimes, especially when you're active, the tiny capillaries inside a person's body will bleed, causing a bruise or pain in the joints. Not dealing with those kinds of bleeds can cause a lot of problems later, including arthritis at a young age. But the good news is, there are good treatments available that can help you manage hemophilia effectively. Treatments that replace the missing factor help your blood clot more like a person who doesn't have hemophilia. These treatments, when used properly, help you keep your bleeding under control so you can live a long, healthy life and follow your dreams without hemophilia ever holding you back. To learn more about hemophilia, visit worldhemophiliaday.org.
With this introductional video, we wanted to start off because it's nicely explained. And in the next slide, I will show you the current treatments, which are based on the regular administration of the clotting factors that are missing. So since the 60s, these clotting factors were isolated from plasma from people donating blood. Unfortunately, this led to the spreading of several diseases within the hemophilia patients group, such as hepatitis and HIV. As shown in the graph here, representing a relatively low safety level for the patient. Within the 1990s, pharma companies managed to produce the clotting factors recombinantly, meaning artificially in the lab, eliminating the risk of spreading diseases. However, the efficacy of the treatment stayed the same. Efficacy of a drug means how good the drug works and how often it needs to be applied to maintain functional levels of the drug. In the case of hemophilia A, 3 applications or injections per week, as shown in the upper graph, are necessary. Of course, you aim to develop a product which can be grouped in the top right of this graph. Very safe for the patient, but also very efficient. New developments are going on to improve the stability of the clotting factors, which should reduce the need for injections from 3 times to twice weekly. A curative treatment would be gene therapy. Gene therapy could cure hemophilia by correction of the not functional clotting factor and enabling the body to produce functional clotting factors by itself and at a sufficient level. Currently, 3 clinical trials are ongoing to correct cells by gene therapy in vivo, meaning directly within the patient's body. As it is hard to control to which cells the viral taxi is transporting its cargo, the genetic code for the missing clotting factor, thus safety aspects need to be considered. The HemAcure approach, however, is different in such a way that we are performing the genetic correction of the cells outside of the body, also called ex vivo. In this way, we can perform safety analysis on the genetic corrected cells before returning them back or transplanting them into the patient. Besides the ex vivo gene correction, we apply a medical device that is called the cell pouch. The cell pouch develops a vascularized environment in which cells can be transplanted and which keeps the cells at this location and prevents them from spreading within the patient's body. This combination sincerely increases the safety benefits for the patient. And how this will work step by step is presented schematically on the next slide by the project coordinator, Dr. Joris Braspenning from University Hospital in Würzburg.
So here's an overview of the HemAcure project shown schematically. The project is set up with different work packages, each responsible for a specific part of the project work. Cell isolation is performed by the team of the University Hospital Würzburg in work package 1. After a brief expansion, the BOEX are shipped to the University of Piemonte, who are responsible for the genetic corrections of the cells in work package 2. After the genetic correction of the cells, the BOEX, now expressing factor 8, are shipped to the University of Loughborough for expansion of cells and subsequent safety studies. After the successful completion of all these steps and processes, the expanded cells are shipped to Canada for transplantations of the cells into the cell pouch by the team of Sernova. To ensure that all these steps are performed in a GMP-compliant way, all processes are guided by Grünewald GmbH. In more detail, all work packages will now be explained by the expert and work package leaders of each work package. So now we will move on with work package 1. Our approach is based on the donation of a certain amount of blood from the patient. As shown in the graph here, blood contains a high number of different cell types. We are interested in a subpopulation, the endothelial progenitor cells, as shown by the red line within the graph. For having the ability to work with them, we need to separate these cells from the other cells, which are available in much higher amounts. And now this is done, I will show in the next slide. To isolate our cells of interest, the endothelial progenitor cells, we use a cell separation fluid. The blood sample is put on the top of the fluid and transferred into a centrifuge. A centrifuge is a device capable of turning objects or samples at a high speed defined as RPM or revolutions per minute. After this high-speed centrifugation, the blood sample is separated into several fractions, as shown here on the right of the slide. Just between the plasma and the cell separation fluid, our cells of interest are collected. This fraction, called PBMCs, stands for peripheral blood mononuclear cells, is containing our endothelial progenitor cells. And this fraction of cells is then transferred from the centrifuge tube into a cell culture flask. The cell culture flask contains growth medium specifically adapted for endothelial cells, meaning that only endothelial cells are able to grow in this medium. An example how these cells look like is shown in the picture here. And these cells are then shipped to our partner UPO for the genetic correction of the cells. So now I hand over to Antonia, who will explain work package 2. Thank you.
Good morning. My name is Antonia Follenzi, and I will explain you the aim of our work package. Once we receive the cells from Würzburg, first of all we put them in culture to amplify, and the next task is to genetically modify these cells with the factor VIII gene. Why this? Because hemophilic patients, they have a mutation in the factor VIII gene, and as a consequence we don't have a production of factor VIII or the production of a mutated form. What we do with these cells once we receive from Würzburg? We prepare lentiviral vectors that are HIV-derived vector. This vector derived from viruses, but depriving the vector from pathogenic genes, we can insert some therapeutic genes, as in the case of factor VIII. Here is, in this design, you can see the correct form of factor VIII that is integrated in lentiviral vectors. In this way, we have a kind of drug that can modify the hemophilic cells. Once the vector is prepared, purified, and titered, is used to transduce or to infect the hemophilic cells. In this way, all the cells will be corrected. What we do with these cells? Before sending these cells to Unilo for amplification, further from amplification, and after to be sent in Canada, we check if the cells are correctly transduced and they secrete factor VIII. And, more important, we do a small proof of concept experiment, injecting some of these cells in a mouse model that we created in the lab that is able to receive human cells. You know, these are immunocompromised mice that can receive human cells, means we can do a xenotransplant. What happened with these cells? First of all, before injecting in the mice, as we said, we checked that the cells were corrected by factor VIII, and so by the insertion of factor VIII by lentiviral vector, and they were able to express factor VIII. You can see here in the immunofluorescent experiment. Once this validation has been performed, we proceed with the so-called in vivo experiment. Means the corrected cells expressing factor VIII are mixed with some microcarrier beads that are kind of little carriers and injected intraportally in our mouse model. And over time, you can see here, from 1 week up to 18 weeks, we will perform APTT assay. Is an assay that allowed us to see if factor VIII is produced and is active. And finally, we perform a blood loss assay that will confirm us that the factor VIII that is secreted is functional. Here you see 4 of the patient's cells that we isolated in collaboration with Würzburg. And after transduction, the cells were injected in the mice. You can see here the time course. That is very nice, because as a proof, we can see that all these mice were able to express and secrete factor VIII with an activity that reached up to 12%. And this activity was present over time up to 13 weeks. After that, it started to decrease. But this is the limitation of this model in the peritoneum. As a confirmation, we saw at the end of our experiment that the blood loss in the corrected mice after cell transduction was acquired. This, for us, is very important because tell us that now these cells are ready to be used by our Canadian partner to be inserted in the cell pouch that they optimize for this project. But before, these cells will be now sent to Unilo that will proceed with the amplification. And with this part, we conclude our results. Thank you very much.
So this is Alexandra Stolzing in University Loughborough. Our role in this consortium is to amplify the cells so that we have enough to treat mice in our experiments or later to treat the patient with multiple of these cells. And in addition, we had to show that during the amplification of these cells, so the mass production of these cells, there were no corrections happening, there were no errors happening, that the cells were safe and functional. So in Loughborough, our job was to take the cells from Italy, amplify them, as you can see in the middle of the slide here. We had to make hundreds and hundreds of plates of these cells, and our job was to see that we can minimize the cost. You have to imagine that this is all very labor-intensive work, and it costs a lot of money to grow these cells and have enough to finally treat actually a patient. So one of our tasks was to find ways of saving money and making it very, very cheaply. So we have roughly indicated what the cost is to grow the cells in a classical way, which could be for one patient up to 1 million euro. However, we found ways of making the cells grow much cheaper. In addition, we had to show that the cells are safe and are not of a threat to the patient. So we had a range of tests that we performed in Loughborough. I have here in the middle of the slide a couple of assays that we were running. First of all, we had to see that the chromosomes, so the identity of the cells, is not changed by the amplification. You have to imagine that the amplification of these cells is a lot of stress for these cells. They're replicating a lot, they're using a lot of energy, and there might be errors in this amplification. So we were checking the karyotype, so the chromosomes, the genetic information of these cells, and we're controlling that no error was introduced into the cells, and the cells were safe to be put back into a patient, in our case, to put back into a mouse model. In addition, what can happen during this amplification is that cells are acquiring a tumorigenic phenotype, meaning that some mutation happens that make the cells unsafe and could potentially induce a tumor in a patient. So with the help of the university in Italy, it was also the testing of the cells in a mouse model where we on purpose injected the mouse with our cells and were checking that they did not form any tumor. This is a very standard safety test. In addition, we also had this test in a dish. So under figure number B, you see on the right side our mouse model, seeing that no tumor is forming, and on the left side our tumor formation test in the dish that both show us these cells are not tumorigenic at all. And in addition, we wanted to show that the cells during the expansion are not losing their ability to produce factor VIII, because this is their important job. And what is also happening during expansion is that the cells are becoming older. So you have to imagine cells like us, when we do a lot of work, are acquiring mutations that are not tumorigenic, but can change the behavior of the cells. So basically, we were also checking a series of phenotypes to show us that the cells are not too old to put back in the body. So once all of this has been checked, we can send the cells off to Canada. And you have to imagine that also we are doing this for every single donor, for every single patient that is donating cells, because every patient gets back his own cells. So we have to show that none of these cells are of any danger to the patient. And with that, I am handing over to our Canadian friends, showing us what happens when the cells leave England and arrive in Canada.
Hello, my name is Dr. Toleikis. I'm President and CEO of Sernova, and I'm representing our Canadian team. The role that we have in this program is to conduct a preclinical safety and efficacy studies of the corrected cells in the cell pouch that were sent to us from Europe. The human corrected cells described above need a safe and contained environment, enabling release of factor VIII into the bloodstream. The cell pouch is a patented medical device that we uniquely designed to provide this safe, vascularized space for the long-term survival and function of therapeutic cells. To conduct the preclinical safety and efficacy studies for the HemAcure program, Sernova manufactured the cell pouch. This manufacturing occurred under strict conditions in a clean room under good manufacturing practices. While the manufacturing process is scalable for human clinical testing, the small sentinel-sized pouch with the therapeutic cells was used for the HemAcure program. The cell pouch is made of materials that have been previously approved for permanent use in the body and has already been shown to be safe and biocompatible in humans for another indication, and is currently being tested in an FDA-cleared human clinical trial in the United States. The next step in the program was to ensure the human-corrected and frozen cells shipped to Sernova from Europe met the quality control criteria developed by the HemAcure consortium members for transplantation. Through defined ship testing procedures, we confirmed that the patient-corrected cells can safely be shipped. Cell batches prepared by the consortium partners and confirmed to meet the established release criteria are shipped to Sernova, where we then establish through repeat testing that the cells still meet the release criteria prior to transplantation. We then developed the surgical techniques for placement of the cell pouch under the skin of the Haemophilia mouse. This resulted in development of instructions for use document for future human clinical trials in the Haemophilia patients. We then confirmed in the Haemophilia mouse model that the development of the cell pouch vascularized tissue chambers that are suitable for cell transplant, similar to our other clinical indication. Having worked through the preclinical preparations, cell pouches were then implanted into the Haemophilia animals, and after development of the vascularized tissue chambers, ship-corrected human cells that met release criteria were transplanted into the cell pouch. These human-corrected cells transplanted into the cell pouch were proven to survive at multiple time points up to the study endpoint at 3 months. Through developed staining techniques, the transplanted human-corrected cells were shown to produce factor VIII. For the first time, we were able to detect some factor VIII in the blood of the Haemophilia mice, confirming that the factor VIII produced in the cell pouch with the cells is able to release the blood into the bloodstream. And this is very important for the program. In a standard tail bleeding test, we also showed that improved clotting of blood in the Haemophilia mouse model relative to the control animals. This first evidence of improved blood clotting with the cell pouch and human-corrected cells compared with untreated Haemophilia mice supports the HemAcure consortium concept that corrected human cells transplanted into the cell pouch can provide and improve clotting for Haemophilia A patients. Now, I'll now turn it on to Martin Zierau for the next part of the presentation.
Hello, Here is Martin Zierau speaking from the Grünewald GmbH. I would like to summarize our contribution to this interesting project. Probably you wonder yourself a little bit, why do I start with a question about spaghetti? What is there similar? But it is clear, every product has to fulfill customer requirements in a high quality for being successful. Spaghetti should be really prepared in a delicious manner if you want to make your guests feel comfortable and be happy. Transduced bioeggs on the other side of the HemAcure project have to fulfill that therapeutical function. The whole setup together with a cell pouch has to be safe for patients. So from this point of view, what is common and what is different? Are there any similarities? Of course there are similarities. In both cases, not only high quality is required, it is required in a constantly stable manner. The quality must be okay if you serve the spaghetti today, tomorrow, or a month later. In the case of treating hemophilia, it's the same. You should be able to treat patient A successfully today, patient B successfully tomorrow, patient C a month later, and of course all the following patients as well. The difference is of course in the hazard of a failing product. Disappointing guests with less tasty spaghettis may be not that harmful. Restaurant owners or probably assess this harm different than those organizing a private party. But in case of bioeggs, if they do not work properly, this is a matter of big concern from a patient perspective. That's exactly the reason why the Grünewald GmbH as a compliance specialist had been involved into that project from the beginning. We had to support the development activities by giving the necessary input for establishing a stable and safe manufacturing process for the bioeggs. Generally, this topic is characterized by three letters GMP. That means good manufacturing practice. What does it mean? Good means that you are able to prevent any harmful events from the patient. In case of HemAcure, for example, from tumorigenicity, or also of course it's a harmful event if you don't have enough factor VIII secretion. From the manufacturing perspective, you have to assure that appropriate rooms and equipment are supplied, efficient process control, professional staff, capable processes, efficient quality control, and efficient process documentation is available. And concerning the practice, of course, you have to realize the requirement by professional and trained staff documenting each step of importance in case of HemAcure. That means, for example, that after each step, as described in this webinar, the isolation of bioeggs, the transduction, the expansion of bioeggs, that after each step you have a quality gate protocol which documents the specifications of the critical attributes. What has to be done in order to put good manufacturing practice into action during the product development? There are three main aspects which can be summarized as the following. Three tasks: qualification, verification, and validation. Qualification means that all the equipment which is needed for producing the transduced bioeggs is qualified. And not only the equipment, but also the testing equipment which is needed for measuring the quality of the transduced bioeggs. Verification means that you check the product characteristics And validation means that you check whether the relationship between the process parameters and the product attributes is okay. From the point of the HemAcure project, it was observed that the properties of the patient are a main characteristic for the performance of the process. That means that we have to document, or we documented each critical parameters at the quality gate protocols, which is the main outcome and which assures that the patients will get a safe and functioning product. So I hand over to the next part of this webinar. Thank you very much.
Thank you, Martin. So what are the future plans for our team? So currently we are seeking for additional funding. Also again, for upcoming European Union framework programs, and in addition, to complete our last basic research experiments. Furthermore, of course, we also will publish our findings in scientific journals. For the long term, of course, we hope the cell pouch with human factor VIII corrected cells, based on our research, will be approved for clinical trials for Hemophilia A.
Okay, thank you very much for the speakers for this nice presentation of your results over the last 3 years. And now, as I mentioned before, we come to the frequently asked questions section. So there were some questions in the course of the event registration that came up. I will go through the questions. And one of the most common question was, is there a therapeutic treatment solely available for Hemophilia A? And maybe the coordinator, Dr. Braspenning, you can answer or give an answer to that question.
Yeah, that's a very good question. The current therapeutic approach, which we are working on in HemAcure project, is focusing solely on Hemophilia A. However, the BOX can also be modified using different genes, and in the future they could be adapted in or corrected in such a way that also BOX from Hemophilia B could be corrected. So the answer is yes, the current treatment is solely for Hemophilia A, but the BOX can in the future be adapted for Hemophilia B as well.
Thank you very much. Another question which comes up regards the cell pouch, and Dr. Toleikis, maybe you can answer. What is the human clinical experience so far in your company with the cell pouch to date?
Yes, thanks very much for the question. Sernova was approved by Health Canada to conduct a clinical study in the cell pouch with cells with another clinical indication. So this shows that the program is already advanced from the clinical perspective. The results show that the pouch itself was safe and biocompatible in the body. Following the transplantation of therapeutic cells, the cells survived in the cell pouch, which is really important, and these became highly vascularized. So in other words, the cells were able to be fed by blood vessels, and that allows the product to be able to be released into the bloodstream. Furthermore, we also showed that the cells in this study were able to produce their product that we were trying to make. And based on these positive results, we've begun a new safety and efficacy study in the United States, which has been cleared by the FDA. So this is the second clinical trial for our cell pouch. This success is helping to pave the way for future clinical studies in Europe for the HemAcure combined ATMP for hemophilia. So this is very, very good progress for our program.
Thank you very much, Dr. Toleikis, for this interesting insight. Another question which was raised, probably Professor Follenzi can answer the best, is how safe is the viral construct that you introduce the correct copy of the factor VIII into the cells?
Yes, lentiviral vectors are integrating vectors and are coming from HIV. But in the last 20 years, a lot of work has been done to make them secure. And first of all, there are several clinical trials ongoing with ex vivo genetic manipulation with lentiviral vectors of hematopoietic cells. Afterwards, these cells have been injected back to the patients. The patients in this moment are cured for their disease, and more important, the integration analysis on the cells that were injected to the patient didn't give any adverse effect. The patients are fine, and no insertional mutagenesis has been observed so far. These are 8 years clinical trials. For sure, we will continue to analyze the cells injected in the cell pouch and to further confirm that a lentiviral vector, even if they are integrating vector, are safe vector for genetic correction. Thank you.
Thank you very much for this answer. Another question which came up regards good manufacturing process. So, Mr. Zierau, what is still missing until a clinical trial can start based on the research of HemAcure?
Despite preclinical investigations are advanced, nevertheless they are not finalized yet. Mostly the results are very promising. It is recommendable to increase the experience concerning the reliability by demonstrating the feasibility at 2 to 3 more patients. Thank you.
Okay, thank you very much. Another question which was raised regards the cells, the expansion, and the safety test. So, Professor Stolzing, what happens if one of the safety tests shows a change in the cells?
We have several steps where the cells have been expanded on a small scale and have been frozen down as a backup. So in case we get corrected cells and have expanded them and we see any kind of change that is unsafe for a patient, we can destroy the cells and go back to one of our backups and restart the process again. So we have the ability because we're not under time constraints, to just go back, repeat the process, do the quality checks, and then see if this is something that came from the patient or is something that was introduced by the process. In case it is introduced by the process, usually this is fixed by just repeating it once more and then showing that the new product is safe, it can be used.
Okay, thank you very much. One additional question which comes up quite prominent is, what about the mild hemophilia patients? So we heard that there are 3 different classes for the hemophilia A and B, and is this approach mainly for the severe or also for the mild patients?
This is Joris Braspenning speaking, and thank you for this interesting question. Initially, we are focusing on the severe hemophilia patients. It's a new therapy approach, so we initially want to treat the severe patients with this new therapy. After collecting some more data with patients, this could be expanded, and also we could start treating mild hemophilia patients. But initially, this treatment is for the severe patients only.
Okay, thank you very much, Dr. Braspenning. So these are the questions that mainly came up in the course of the registration. So there was one additional question via the question and answer section. So probably everybody is stunned by the results of the project, but maybe this one question I can ask, and it refers to the cell pouch. So the question was, what happens with the device after 3 months in the body? And maybe Dr. Toleikis, you want to answer that question.
Yes, thanks very much for that question. So there are a couple of points here. First of all the materials for the cell pouch have already been approved for permanent use in the body, so we know that those materials are safe for the patients for long term. Secondly, we have conducted a large animal preclinical study showing that the cell pouch is safe all the way through one whole year of the study, and this is considered to be a very, very long-term study for a preclinical study. We've also done a large animal model with cells in the cell pouch for almost a year that also showed that the cells are surviving and that the device is working. So again, this is considered to be long-term. And finally, in our patient study, we have shown in a patient that the cell pouch has been in the body for longer than a year and safe from that perspective too. So all of these point to the direction that the cell pouch is very safe and biocompatible long-term. And a one-year study would be considered very, very positive. We did not get any fibrosis in the device or have any issues whatsoever from the safety perspective from that during this time.
Okay, thank you very much for that answer. So we're heading to the end of this webinar, and once again I want to mention that even if you do not have a question now, but maybe later on, the web page of HemAcure and the Twitter account is still online also when the project ends. Unfortunately, this is next Wednesday. So when you think about the project and you still have questions, you can just write us an email or a tweet, and we come back to you. And then, of course, the webinar is recorded, and we will upload this to the homepage and the Arctic's YouTube channel. And all the questions that were raised, we will put up and will be raised maybe later, will be in a written follow-up, which we also will put online. So finally, I'd like to thank all the speakers today and the Arctic's communication team for setting up this webinar. And of course, I would like to thank our audience. Thank you for taking the time today. Excuse a little delay in the beginning. And we highly appreciate the interest in our research. Have a nice day and goodbye.